Good morning, students. I hope you all are fine. So, today we are going to start from the chapter number 6, which is Molecular Basis of Inheritance. So, guys, this chapter is all about the genetic material and gene expression. So, do you know which type of substances are known as the genetic material? Yes, you know. These are the nucleic acid. So, nucleic acids are known as the genetic material. So, first, you should know what are the properties or characters one genetic material has. So here see, these are the properties of genetic material. That means always remember, to become a genetic material, one specific substance must have some specific properties like these four. Here the number one, see, it should replicate. The first property of genetic material is it should replicate. That means it should have the ability to produce its own replica. Do you get my point? It should replicate means what? It should have the ability to produce its own replica. Replica means exact copy. When some substance can produce its exact copy, then this is called the property of genetic material. Okay. So now the second one. It should be chemically and structurally stable. Number 3, it should not undergo frequent changes, which is called mutation, that are necessary for evolution. So you all know the mutations are very important for the evolution process. But if these genetic materials frequently change their structure, then it will lead to some disease but not evolution. Okay, that is why it should not go, that means undergo frequent changes. The number 4, it should be able to express itself as Mendelian factor. So who was Mendel? Anybody? Who was Mendel? So Mendel, there are three very important and famous laws proposed by Mendel, which are known as the laws of the inheritance. Mendel's laws of inheritance. So he is known as the father of genetics. Remember? So Mendelian is known as the father of genetics. So to become a genetic material, one substance must or it should be able to express itself as Mendelian factor. So these are some basic property of genetic material. Okay, remember this point. These four are the basic important properties of a genetic material. So now the next one. So here see, the round structure is the cell, and inside the cell, that is the nucleus. And you know, all the DNAs, chromosomes, all these genetic materials are found inside the nucleus. So this one is the chromosome. Okay, that X-shaped structure is the chromosome, and all the chromosomes contain some chromatin network. So these long thread-like structures are the DNA here. All these are DNA, and in the DNA here see some round structures. These are called nucleosome. Okay, so around the nucleosome, these are the DNA strand. So this is the DNA strand. So that means all these nucleosome structures are found in the chromosome. So what is nucleosome? I will discuss it in our later class. So now move to the next slide. Next one is the discovery of nucleic acid. So what is nucleic acid? You got this one in your first year. What is nucleic acid? Nucleic acids are made up of the nucleotides. So what are the nucleotides? You know, nucleotides are the monosaccharide. These are the micromolecule, not monosaccharide. These are the micromolecules, the small molecules present in the living organisms. Then one example of the macromolecule, that is the nucleic acid. So nucleic acids are made up of the micromolecules called nucleotides. Okay, so these nucleic acids were first discovered by the Friedrich Miescher in 1869 from the nucleus of the pus cells. What is pus cells? Okay, Friedrich Miescher in 1869 discovered the nucleic acid from the nucleus of the pus cells. You know what is pus cells? Pus cells means when some antigens means you can say any bacteria, any virus, when some disease causing agents enter inside our body, then these are killed or destroyed by the WBC. After destruction, all these cells are known as the past cells. Okay, all these destroyed cells are known as the past cells. So Friedrich Mitchell discovered the nucleic acid from the nucleus of these destroyed past cells, and these were found in the waste surgical bandages and named these substances as the nuclein. But later the scientist Allman named it as the nucleic acid. Why? Because it was sorry because it shows some acidic properties. Okay, these substances which were named as the nuclein, they show some acidic property, and that is why later this nuclein were named as nucleic acid by Altman. Okay, this is the discovery of the nucleic acid. So now come to the next slide, that is nucleic acid, the structure of nucleic acid. So you know the nucleic acid is made up of the nucleotides, and these nucleotides are made up of three chemical components. So before that, here see, there are two types of nucleic acid. One is called as the RNA ribonucleic acid, which is only found in some viruses, okay, in some other organisms also, but mostly in the viruses. And one is called as the DNA, which is the double stranded, and this is found in the eukaryotic cell. Then next, nucleic acid consists of a large number of building blocks called nucleotides. So remember, there are two substances. One is called nucleoside, one is called nucleotide. What is the difference between the nucleoside and nucleotide? So here see in the diagram. Here see the nucleoside, the orange color ring. So this is the structure of nucleoside, where the orange ring is the nitrogenous base and the yellow one is the pentose sugar. Okay, here see in the nucleotide, the purple colored ring is the nitrogenous base, the lilac color or slight uh, pinkish color is called as the pentose sugar, or the last one is the phosphate group. Okay, that means nucleoside consists of nitrogenous base and pentose sugar, and nucleotides consist of nitrogenous base, pentose sugar, and phosphate group or phosphoric acid. So these are the difference between the nucleoside and nucleotide. So here, this is one example. So here say the green color ring, which is one nitrogenous base called adenine. So adenine binds to the pentose sugar. When the adenine binds to pentose sugar, this combination is called as the adenosine nucleoside. Okay, this is called as the adenosine, which is the nucleoside. So now when this nucleoside binds to the phosphate group, it will become the nucleotide. Okay, yes, see, the green one is the nitrogenous base, then it binds to the pentose sugar and form the adenosine, which is one example of nucleoside. Now this combination bind to the phosphate group and form the nucleotide, which is called as the adenosine monophosphate. Okay, so come to the next slide. Now nucleotides. So I have told you already that nucleotides consist of three chemical components. One is called nitrogenous base. You got this one in your first year course. So second one is the pentose sugar. There are two pentose sugar. One is ribose, which is present in the RNA. One is deoxyribose, which is present in DNA. And last one is the phosphoric acid, or you can say phosphate group. Here see in the diagram the nitrogenous base. There are how many nitrogenous bases? There are 5 nitrogenous bases: A, T, G, C or U: adenine, thymine, cytosine, guanine and uracil. Okay, when the nitrogenous bases bind to the pentose sugar, here see the green color ring is the pentose sugar, and on the side this is the phosphate group. So this way all these three components bind to each other in the DNA structure. So after that, come to the next slide, which is the types of nitrogenous bases. So there are two types of nitrogenous bases. One is purine and one is pyrimidine. Look at the diagram. The purine is double ring structure. Okay, the purine is double ring structure, whereas the pyrimidine is single ring structure. Here see, the purine is double ring structure, pyrimidine is single ring structure, and there are two types of the purine, adenine and guanine, and three different types of the pyrimidine, cytosine, thymine and uracil. So this is the different types of nitrogenous bases. So now come to the next slide. So here see, there are two types of sugar, pentose sugar. Pentose sugar means what? 5 carbon sugar. In the ring there are 5 carbon present, and that is why these are known as the pentose sugar. So there are two types of pentose sugar. One is called as the deoxyribose, the first one in the diagram, the deoxyribose, which is only present in the DNA, and that is why the full form of DNA is deoxyribose sugar. And here in the second diagram, this is the structure of ribose sugar, which is present in RNA. So what is the difference between the deoxyribose and ribose? In the diagram, see the second number position. In the ring, see the second number position. In the deoxyribose sugar, in the second number position, one oxygen is missing. Can you see that? In the first diagram, in the second number position, one oxygen is missing. Whereas in the second number picture, in the second number position, the oxygen is present. And that is the difference between the deoxyribose sugar and the ribose sugar. Okay. So these are the pentose sugar present in DNA and RNA. Next one, the backbone of nucleic acid. This is the backbone of nucleic acid. Backbone means, that means the two strands of nucleic acid, two strands of DNA simply. So you already know that the DNA consists of two polynucleotide strands. Okay, these two polynucleotide strands are called as the backbone of the DNA. Okay, so here see, the phosphoric acid or phosphate binds to sugar. Sugar means pentose sugar. Always in these two strands, here see in the diagram, all these round circle, red round circles are the bonding between the phosphate group and sugar. Okay, all these round, the red color, round red circles are the bonding between, the bond between the phosphate group and the pentose sugar. Okay, and this is the backbone of the DNA, just single backbone this is. Okay, this way another backbone is also present in the DNA structure, because DNA made up of two polynucleotide strand. So next one, phosphodiester bond. So what is phosphodiester bond? Phosphodiester bond meaning, in one strand of DNA, the phosphate group binds to the pentose sugar with the help of phosphodiester bond. Remember, in one strand of DNA, the phosphate group binds to the pentose sugar with the help of the phosphodiester bond. So you can see this all red circles are the phosphodiester bond, which is the bond between the pentose sugar. All these green are the pentose sugar. In the diagram, all the green colored ring are the pentose sugar, and all the round, small round structures are the phosphate group, and all the yellow colored ring are the nitrogenous base. Okay, so in the diagram, see the bond between the small round and the green color ring. All these bonds are known as the phosphodiester bond. Okay. So next, see. So this way, all these different types of chemical components are found in the DNA. That means the first one is the nitrogenous base. The second one, the dark blue, is the sugar, and here this is the phosphoric acid. Okay, always the phosphoric acid or phosphate group binds to the sugar, whereas the sugar binds to the nitrogenous base. So pairing of nucleotides. So how these nitrogenous bases pair to each other, bind to each other in the DNA molecule. So here see, always the purine binds to pyrimidine. Okay, there are two types of nitrogenous bases. One is called purine, one is called pyrimidine. So remember, always the purine nitrogenous bases bind to the pyrimidine nitrogenous bases, and I have already told you that the purines are double ring structure and pyrimidines are single ring structure. Okay, so look at the diagram. All these double ring structure inside this DNA molecule, all this double ring structure, one is the green color, one is the orange color. This way, in between this structure, these are the purine. Okay, all these singles are the pyrimidine inside the structure, and outside the structure, these are the phosphate group and the sugar. Okay, remember, Inside the structure, these are the nitrogenous bases. The double ring structures are the purine and single ring structures are the pyrimidine. Okay, and the outside, in the outline of the structure, these two are, one is the phosphate group, one is the sugar. The phosphate group and the sugar bind to each other with the help of phosphodiester bond. Okay, and this sugar bind to the nitrogenous bases with the help of the glycosidic bond. Remember, the phosphate group binds to the pentose sugar with the help of phosphodiester bond, whereas the sugar binds to the nitrogenous bases with the help of glycosidic bond. And remember, in a DNA structure, always purine binds to pyrimidine. Okay, not purine, not pyrimidine, but always purine binds to pyrimidine. And purine, here see there are two examples here. In the DNA structure, if one strand contains the adenine, then the second strand must contain the thymine. That means adenine always binds to the thymine and guanine always binds to the cytosine. Okay? Adenine always binds to the thymine by 2 hydrogen bond and guanine always binds to the cytosine by 3 hydrogen bond. So this is the pairing of nucleotides in the DNA structure. Remember, in the two strands of DNA, the phosphoric acid binds to the sugar with the help of the phosphodiester bond, which is called as the backbone of the DNA. And inside the DNA structure, the sugar binds to the nitrogenous bases with the help of glycosidic bond. And all the nitrogenous bases bind to each other with the help of the hydrogen bond. So I will tell you all these different types of bonds in our next class, which is all about the structure of double helical model of DNA. Okay. So this is all about the discovery of nucleic acid, the properties of suppose genetic material. Okay, so next class we will discuss about the structure of DNA and all the different types of RNA and their function. Okay, till then, bye bye and take care.